Good talk learners, welcome to today's life sciences lesson. My name is Vitoro Makwala. This program is brought to you by the Northern Cape Department of Education in collaboration with Pagama Research and Development. So today's lesson, we are still looking at our paper 1 revision. So we are going to focus on some of our past papers as we have been doing previously. So we are just going to be looking at section B of our past papers. So here we will be looking at our question 2 part, and then later on we will then look at our question 3 part. Remember, our paper, our life sciences paper is out of 150, as we can see here, with the topics as well as their mark allocation together with their percentages. We have for term 1, reproduction in vertebrates, human reproduction. With reproduction in vertebrates, we have 8 marks, whereas human reproduction, we have 41 marks, as we can see here. And then in the second term, we have responding to the environment in humans, whereby we would be looking at things such as the human brain. We will be looking at the human eye, the human ear, and how humans would then respond to the environment through these different parts of the body. Right, and then we have responding to the environment in plants, where we would focus on plant growth hormones, mainly with the auxins and the gibberellins. Right, and then lastly, we have the endocrine and homeostasis in humans, where we will be looking at negative feedback mechanisms in order for humans to be able to maintain homeostasis within their bodies. Right, so responding to the environment in humans is 54 marks out of the 150. Responding to the environment in plants would be 13 marks, very small marks there. And then lastly, the endocrine and homeostasis in humans, which is 34 marks. So now, of these 150 marks, just a reminder, we have our section A, which has 50 marks, which we would normally have your multiple choice questions. You would have your biological terms, and then you would also have your column A, column B, where you would choose your terminologies based on the description that you have been given, right? And then you would usually have 1.4 and 1.6, which are usually some short questions, which would then make up your 50 marks for section A, right? All right, so in our section B, we have question 2 and question 3, right? So both questions have 50 marks each, meaning that section A 50 marks, question 2 50 marks, and question 3 50 marks. So here, today we will only be looking at the 50 marks that makes up our question 2, right? So we are going to focus on all of those questions that could possibly be in the exam for paper 1, right? And then in the next lesson, we will look at question 3, which again would then just sum up what a paper 1 exam would look like, right? So here we will be looking at all of these different topics that we've just mentioned on the previous slide, right? So all of these questions would possibly be part of the questions that we are going to look at in both question 2 and question 3, right? So by the end of our lesson today, we should be able to label and give functions of the parts of the male reproductive system. So we look at the male reproductive system, we will look at the different parts that make up the system, as well as their functions, right? We should also be able to describe the functioning of the human eye, right? So here we will look at the processes that are involved in the human eye. So we would look at things like pupillary mechanism, we would look at things like accommodation, right? So those are the processes, right? Now remember, when you are looking at these processes, you should also be able to label the part, right? The human eye. You should be able to label the human eye and give functions. By you being able to give functions, it means that you would simply be able to describe these processes that are involved, because it is the different parts that are involved, and the functioning of those parts essentially make up the processes, right? You should also be able to differentiate between the sensory and the motor neuron. So you should know what the motor neuron does, you should know the direction of the motor neuron, as well as what the sensory neuron does and its direction as well, right? And then with that, you would then be able to draw a reflex arc, right? So you should be able to draw a labeled diagram of the reflex arc, whereby you are going to be able to show all of these things: the motor neuron, the connector neuron, the sensory neuron as well, and just to be able to show which part of the system does the sensory neuron go through, so your ventral root and the dorsal root, and be able to just differentiate that, right? So we are going to look at that, and you should also be able to describe the functioning of the human ear. When we look at the functioning of the human ear, we would either be looking at hearing or balance, right? So you should be able to know the different parts of the ear, which will then be able to assist in answering questions on functioning of the human ear, should it be hearing or in balance as well. Right, so let's just get to our first question, which is question 2.1, whereby it says the graph shows changes in the diameter of the pupil under different luminescences. Luminescence refers to how bright light is. So that simply explains to us what we are looking at when we are looking at the different luminescence, right? So this is the brightness of light, basically. The diameter of the pupil ranges from approximately 2 millimeter to 8 millimeters, right? So it means that here we are going to be seeing the diameter of the eye or the pupil as the luminescence changes, or as the brightness of the light changes. So if we have a look at the graph that we have been given here, we have our luminescence on the x-axis and the diameter of the pupil on the y-axis. This essentially tells us that the luminescence controls the diameter of the eye, meaning that as the luminescence changes, it then causes changes in the diameter of the eye or the pupil, right? So our first question says it's 2.1.1. Give the diameter of the pupil at point A. So let's go back and look at point A. Point A is what we have here. So this simply just needs you to have a ruler and then just check what would be the luminescence, not the luminescence, but the diameter, what the diameter of the eye would be at point A. So the luminescence is 0.1 and the diameter is 6 millimeter. The question is asking for the diameter, so our answer here would then be 6 millimeter. Right. Okay, let's go back to the second question, 2.1.2. It says calculate the average diameter of the pupil using only points indicated between point A and B in the graph shown. Show all working. So here we are calculating the average diameter using the points between A and B, only the points between A and B. Now let's have a look at the points between A and B. We only have 4.9 and 3.8, right? So this simply means that you are going to say 4.9 plus 3.8 divided by 2. Every time you get your average, you will add the different values that you have, and then you will divide by the total number of values. What this means is that if we were adding all these points, which is 1, 2, 3, 4, 5, 6, 7, it means that we were going to add all of these points. So we would say 7.7 plus 7.5 until 2.1. We add all of them, divide by 7, because we have 7 points. However, in this case, we only have 2 points. It is the 4.9 plus the 3.8. Right. So all of that divided by 2, you simply punch that into your calculator and you would then get that final mark, that final answer, and that is how you get the mark. Now let's have a look at the mark allocation here. It is 3 marks, meaning that it would be possibly your addition, the division by 2, and then your final answer, and then your addition, division by 2, and the final answer. So it is 4.9 plus 3.8, that gives us 8.7. Then the 8.7 divide by 2, like I said, dividing by 2 is a mark, and then finally your 4.35 millimeter, which would be your final answer. Again, remember, every time you are doing calculations, if you've been provided with a unit, make sure that even in your answer you do indicate the unit that you were given. So here it will be 4.35 millimeters. That is how you would score your 3 marks. Alright, and then the next question, 2.1.3. Describe the relationship between luminescence and the diameter of the pupil from point A to point B. Right, now let's go back and have a look at what is happening here. So we can see that the diameter is basically decreasing from the first point until the last point. There is that constant decrease. However, this decrease is happening as we are experiencing an increase in the luminescence, meaning that when you describe the relationship between two things, in this case, you're going to explain what happens to one variable as the other variable changes, which is usually the relationship that you should be able to see between a dependent variable and an independent variable. Speaking of which, by simply looking at this graph, you should be able to tell us which one is the dependent variable and which one is the independent variable, right? So here we can see that as the luminescence increases, with the increase in luminescence, there is a decrease in the diameter of the pupil. So that is the relationship that you would give, that as luminescence increases, the diameter of the pupil decreases. Now, by that, you can also be able to identify that the luminescence is our independent variable and the diameter of the pupil is our dependent variable, because the change in luminescence is what causes a change in the diameter of the pupil. Now, which will then also take you to having that understanding of how the eye works, right? Remember, as light increases, or how as the brightness of light increases, your pupil would constrict, right? As your pupil constricts, it means that the diameter of your pupil will become smaller and smaller. Your diameter will decrease. Why? This is to decrease the amount of light entering the eye in order to protect the eye, right? So this is why the luminescence, an increase in the luminescence leads to a decrease in the diameter of the pupil. Now, this doesn't end there. As you are understanding what causes a change in the luminescence causes a change in the diameter of the pupil, you should also understand the different parts that are involved and describe that process, which would probably be in the next coming questions. So here, as the luminescence increases, the diameter of the pupil decreases. Right, that is your 2 marks. Right, and then our next question, it says, which is 2.1.4, explain the role that pupillary reflex plays in changing the diameter of the pupil from point A to point B, which is what I was just saying now. So, what role does pupillary reflex play in the changing of the diameter? What role does the pupillary reflex or pupillary mechanism play in making sure that the pupil dilates or the pupil constricts based on the changes in the luminescence? Here we are looking at constriction of the pupil diameter becoming smaller in order for the light entering the eye to be less, meaning that you must describe all of that before you can even get to the point of saying the pupil will constrict and the amount of light entering the eye will decrease, right? You must know which muscles are involved in, right? So firstly, this is in the iris, right? So you can say the circular muscles of the iris contract, or you can say in the iris, the circular muscles contract, right? So by mentioning that this is taking place in the iris, you do get a mark. And by mentioning that the circular muscles contract, you also get a mark. Now, do not confuse the ciliary muscles and the circular muscles. We speak of the ciliary muscles in accommodation, and in pupillary mechanism, we speak of the circular muscles which contract, right? And then the radial muscles relax. Radial muscles relax, circular muscles contract. Now, an easy way to remember this, it would be that the circular, which start with a C, contract, CC. The radial muscles, which start with R, relax. So, for pupillary mechanism in bright light, this is pupillary mechanism in bright light. Why do we say it's in bright light? Because the brightness of the light keeps increasing, the luminescence keeps increasing, which is very important that they have told us what luminescence means. We were told here what luminescence is. Luminescence is how bright light is, meaning that as luminescence increases, the brightness of the light increases, right? So the radial muscles relax, which will then cause the diameter of the pupil to decrease, or it will cause the pupil to constrict, right? Diameter decreases. When the diameter decreases, then you know that it means pupil constricts. Diameter decreases, and then what happens last? Less light will enter the eye. Obviously, you would already know that as the diameter becomes smaller, there isn't enough space for more light to enter. Therefore, the amount of light entering the eye would have decreased, and that is how you will score your 3 marks. Or you can even score more than 3 marks. So you have the first mark here 1, 2, 3, 4, 5. So you can even get more than 5 marks. So just be able to describe this as it is, and then you'll be able to score your marks. Now, when you are looking at this process at dim light, it is basically the opposite. The circular muscles will relax, the radial muscles will contract, the diameter of the pupil will increase, or the pupil will dilate, it will become bigger. Why does it become bigger? There isn't enough light entering the eye. So as the diameter increases, it is to allow more light into the eye. And what would happen then? More light will enter the eye. And that is how you would then score your marks. So if you understand pupillary mechanism in bright light, you don't even have to spend a lot of time trying to understand the dim light, because it is just the direct opposite of dim light. Right, 2.1.5, it says define each of the following: luminescence and reflex action. I think for part A there, that is just a free mark, because we've already been given the definition or the description of luminescence. We know that this is how bright light is. Simple as that. And then you get your mark, right? So it takes us back to reading your questions or the information provided carefully. When you get your question, do not just jump to 2.1.1. Read that information, understand what that information tells you, underline that information. Whatever you think is important, just underline it, go through it again just to get a better understanding, because you will then be able to score simple marks like what you are seeing with 2.1.5a. And then 2.1.5b, a reflex arc. So reflex action, not a reflex arc. We know that when we're talking about reflex action, we're talking about quick automatic responses to a stimulus, right? So this is a response that is very fast to any stimulus in order to protect the body from being harmed, right? So it is a quick, involuntary, automatic response to a stimulus. You do not volunteer to respond in this case. When we talk about reflex actions, we are basically talking about situations where you would have placed your hand on a hot surface, you would have walked into a sharp nail, right? So those are responses that you would take, or responses that would happen very quickly as there is that stimulus that you would have to have reacted to. Now 2.1.6, it says the beam light from the laser pointer has a high enough luminescence to burn any receptor cell in the retina in about 10 seconds. Explain how shining the laser pointer directly into the retina for more than 10 seconds will affect your eyesight. Now, if they are saying it can burn any receptor, it means that should this laser be directed to the retina for more than 10 seconds, it would then burn these receptors. Now, without receptors, no stimulus from the external environment can be perceived. It means that if the receptors in the retina, the rods and the cones, are bent, it means that no stimulus from the external environment would be received. No light will be received. Now, if no stimulus is received, it means that there is nothing that is going to be converted into an impulse in order to be transmitted to the cerebrum for interpretation. If that does not happen, it means that this person would have lost their eyesight. This person will not be able to see. Therefore, your vision will be impaired, right? So the cones or the rods, or you can say the photoreceptors, will be damaged, as they said, they will be bent, right? So no stimulus will be picked up. There are no receptors to receive the stimulus. Therefore, no impulse will be created. No impulse will be transmitted to the cerebrum through the optic nerve. Remember, the impulses are transmitted through the optic nerve, and therefore vision will be impaired. This individual will not be able to see as the receptors are damaged. Now with 2.2, we are still looking at that eye. Now let's just start by trying to label what we have here. We have the ciliary muscles and the suspensory ligaments. I think once we start seeing these, we can think of processes such as accommodation, because these are the muscles that are involved in accommodation. However, let's just have a look at our questions and see what they have for us. The diagram shows the internal structure of the eye. Identify the structures labeled A and B. We've already identified those. Because you've already identified these, you're simply just going to write down your answers without having to worry too much about that. So that is the ciliary muscles as well as the suspensory ligament. Right. And then 2.2.2, the optic nerve is a sensory neuron. It is a sensory neuron because it transmits impulses to the brain or to the cerebrum. Tabulate two differences between the sensory neuron and the motor neuron. Right, like I just said, sensory neuron transmits impulses to the brain. Motor neuron transmits impulses from the brain to the effectors. This one from the receptors to the brain, motor neuron from the brain to the effectors. Right. Now we can also look at the structure. Okay, let's just have a look. Impulses towards the central nervous system. Yes, we've looked at that. Motor neuron carries impulses away from the central nervous system, and then it has only one dendrite. So it is what we say unipolar, with only just one dendrite, whereas the motor neuron has many dendrites. Right. Now, with the sensory neuron, both the dendrite and the axon are myelinated, meaning that they are surrounded by the myelin sheath. Whereas with the motor neuron, it is only the axon that is surrounded by the myelin sheath, right? And then next we have the fact that it makes up only part of the white matter, that is the sensory neuron, whereas the motor neuron makes up both the white matter and the gray matter. And then found in the dorsal root. Remember, when you draw your reflex arc, you should be able to indicate this part. Why is it found in the dorsal root? Remember, this is based on the position of the cell body. In the dorsal root, we have the dorsal root ganglion, which shows that it accommodates that centrally located cell body of the sensory neuron, which is not the case with the ventral root. It doesn't have a ganglion because the motor neuron does not have that centrally located cell body. Lastly, the sensory neuron has a shorter neuron, whereas the motor neuron has a longer neuron. Now, let's have a look at the marks. The question said, how many differences? 2 differences. But how many marks do we have? 5 marks. Now, the last mark is for tabulating. Now, this means that if you do not draw a proper table, you will lose a mark for that table. Simply drawing a cross like this is not drawing a table. Draw a proper table. Indicate that on this side we are talking about the sensory neuron, this side we are talking about the motor neuron, and then have each point shown specifically to show that here we are talking about the impulses, and on the other side as well the impulses. When we are talking about the dorsal root and the ventral root, it should be like that. The length of the axon on both sides should be indicated. So you do get that free mark for simply drawing a table. It means that even if you do not know the answer to this, you do not know the differences between the motor neuron and the sensory neuron, you simply draw that table and then you will get your 1 mark. Right. So do not lose marks because of small things. Make sure that you focus on such small things and make sure that you score those marks, even if maybe you might not know the full answers to some of these questions. However, you should be able to answer these questions as well. 2.2.3, it says name the part of the nervous system which is composed of the cranial and the spinal nerve. So here, when we are looking at anything that is outside of the central nervous system, we are then looking at the peripheral nervous system. So the peripheral nervous system is made up of the cranial and the spinal nerves. Okay, 2.2.4, describe how structures A and B, the suspensory ligament and the ciliary muscles, enable the ability to focus on objects further than 6 meters away. Exactly. Now we are looking at accommodation. Right. This question did not outright say describe the process of accommodation when a person is looking at an object that is 6 meters away. However, once you start seeing that a person is looking at an object and this object is at a certain distance, you know that we are looking at accommodation. The same way, when you are looking at a question and it's talking about light, then and the diameter of the eye, then you know that. So just like when you are looking at bright light and the diameter of the eye, you know that we are looking at pupillary mechanism. So you should not expect every question to be straightforward. You should not expect every question to say describe accommodation, describe pupillary mechanism. You should be able to read into that question and understand what is required of you. So here we are expected to describe accommodation at a distance 6 meters away. Now you should also understand, if it is 6 meters away, is this near vision? If it's less than 6 meters away, is it near vision or distant vision? Again, we're not told. It means that you should be able to understand that. Right. Okay. So here you can mention what structure A is, or just say the ciliary muscle, or just say structure A, structure B. It doesn't really matter because you've already named them previously. However, it is easier to name them so that you can just have that flow in your answer. Right. So firstly, your ciliary muscles would relax. Right. Now when your ciliary muscles relax, they are going to cause the suspensory ligaments to become taut, meaning that your suspensory ligaments are going to be tight, they're going to be pulled. Right. So it means that if we have a look at this part here of the suspensory ligament, it's going to be pulled. Whereas if it is not pulled, you would see that it would be having a wavy like structure like that. So in this case, suspensory ligaments have been pulled, they are tight. Right. Now when the suspensory ligaments are pulled like that, it means that they are also pulling on the lens of the eye. Right. If the lens is pulled like that, we can talk about two things in terms of the lens. The tension on the lens is going to increase because it has been pulled, right? And the convexity of the lens is going to be less. The lens is going to be less convex or less round or more flat. Right. When this happens, remember the convexity of the lens now controls how much light will be refracted. Right. If the lens is less convex, it means that it's not big enough or it's not too big, therefore it does not bend the light rays more. The refraction will be less. The refractive power would decrease. Right. If the refractive power has decreased, it means that now the light rays will then be able to land on the retina. Right. Let's have a look at that. Ciliary muscles have relaxed. The suspensory ligaments are taut or tight. Right. The tension would increase on the lens because it has been pulled. Now, if the tension increases, the lens is less convex or flatter, more flat, right? And then the refractive power would also decrease, meaning that the light rays would be bent less or refracted less, allowing light to fall onto the retina or a clear image to be formed on the retina. Right. So you can also mention that a clear image is formed at the retina. Sometimes they do give a mark for that, right? So this is how you would explain accommodation. Okay, now let's have a look at the next question, 2.3. We are given a structure of the human brain. The diagram shows the section through the human brain. What are we given here? The corpus callosum, the cerebellum, spinal cord, the pons, as well as the medulla oblongata. Right. Now, first question 2.3.1. Give one function of each of the following: the function of the corpus callosum, the function of the cerebrum, as well as the function of the spinal cord. Only one function per part, right? So with the corpus callosum, which is what we are seeing here, This is the part that is responsible for just having that separation or connection between the two hemispheres of the cerebrum. The cerebrum, remember, is divided into two hemispheres. So it joins the two hemispheres together, the two hemispheres of the cerebrum together, and it allows for communication between those two hemispheres. Right. Okay. So you're able to see that where there's this part, that is where there's the right hemisphere on the other side and the left hemisphere on the other side. So only one mark here. So you can either say it joins the two hemispheres or it allows for communication between the two hemispheres. You don't have to give both answers because why? It is only one mark. So do not waste your time giving two answers. Right. And then the cerebrum, we know that the cerebrum is responsible for receiving all sensations. We know that it is responsible for voluntary actions, things like talking, moving, running, and so on, right? So receiving or interpreting all these voluntary actions or all sensations, and it is also responsible for memory, recalling, and high thought processes, right? So controls voluntary actions, receives and interprets sensations from sense organs, as well as high thought processes. Again, here one mark, so you can give any of these, not all of them, but only one, right? And then lastly, we have the spinal cord. The spinal cord is responsible for transmitting impulses between the peripheral nerves and the brain, or between the brain and the effectors, right? Again, it allows for reflex actions to take place. Why do we say it allows for reflex actions to take place? Is because during a reflex action, the impulses do not necessarily reach the brain. Impulses are sent to the spinal cord and a reaction takes place without having to have an impulse being directed to the brain specifically. 2.3.2. Draw a labeled diagram of a simple reflex arc and indicate the direction in which the impulse travels. Right. So when we're talking about a reflex arc now, we are going to show our motor neuron, we are going to show our sensory neuron, we are going to show the root, the dorsal root and the ventral root. We are going to show our connector neuron as well. And remember, when you draw this, it can either be on one side or the left side or the right side, whichever one you choose to draw, as long as it is clear which neuron is which. Now let's have a look at that. Right. Number one, a caption or a heading for your drawing. Every drawing must have a caption. Every drawing must have a heading. So already you have scored yourself 1 mark for that. Right. So even if your drawing is wrong, for that it's a mark. Right. Now we have our receptors. Remember, our receptors are the ones that are receiving the stimulus. Now from there you must show where is this impulse going. Remember, the receptor is going to convert the stimulus into an impulse. Now the impulse is traveling towards the brain. There we have the arrows indicating the direction of flow, right? We have our sensory neuron. Indicate that this is a sensory neuron. Show your centrally located cell body. Show the dorsal root or the dorsal root ganglion. And then now we are inside the spinal cord or the central nervous system. Show that we have the white matter and the green matter. White matter on the outside, green matter on the inside, which is something that we usually discuss as we start by looking at the structure of the brain, whereby we look at which parts have the white matter and which parts have the green matter, right? Which is something that you should also be able to indicate when you are looking at the cerebrum and the cerebellum, right? So white matter on the outside, grey matter on the inside. Now, once you reach the central nervous system, indicate that we have the connector neuron, right? So where the motor neuron and sensory neuron ends, you can show the connector neuron. You can also show the synapse over there, right? And then from there you can show the motor neuron, which leaves the central nervous system via the dorsal root, the ventral root. Indicate your ventral root, indicate the direction again, that from here goes back to the effectors, right? So we have indicated the motor neuron, the ventral neuron, the interneuron, and both the gray and the white matter. Now this is a complete diagram of your reflex arc. Now this can be from this direction, whereby you would have shown all of this in that direction, your central nervous system, and then in that direction again. So it doesn't really matter which side you draw it from, whichever side that you would have preferred. Right, now let's have a look at the criteria for matching this. When you have this criteria, you must follow it, right? You will not be given this criteria in the exam. You must just know what are the things that are expected in this in these drawings, and then from there you will know which criteria to follow in order to be able to score all your marks. The first one says the neuron positioned correctly in the diagram. Are your neurons positioned correctly? What we mean here is that you should not have your sensory neuron in the ventral root and your motor neuron in the dorsal root. That would mean that your neurons are not placed correctly. Right. And then the second one, the correct direction of impulses. Do you indicate the direction of impulses? Yes, we have indicated the direction here, that from the receptor to the brain, that way, from the brain to the spinal cord actually, and then from the spinal cord to the effector, that way. Right, and then next, any two correct labels. We have so many labels here. You can even get some labels wrong. However, if you only have two that are correct, then you're sorted. If your motor neuron is there, your sensory neuron is there, your interneuron is there, you already have your 2 marks. If your effector or receptors are there, you already have your 2 marks. You might not even show your white matter and your gray matter. You might not even show your dorsal root and your ventral root. However, if these main ones are there, then you have your 2 marks. But it is also important just to have a good understanding of the whole diagram. Right. Last one, the caption. As I said, first thing first, have your caption. It is somehow of a free mark. So make sure that you have that. Okay. So that is what we have for the diagram. So the diagram may be drawn on the left side or on the right side of the spinal cord. It really doesn't matter which side you draw it. Okay, now we have 2.4. The diagram shows the internal structure of the ear. Right, like I've been saying, when you are looking at these parts, be able to label the part, be able to give functions for the different parts, and therefore you will have a good flow of when answering these questions, because you already know what is this part, what does this part do, and so on. Then you will easily be able to answer any question that you get. Right, now let's have a look at this diagram here. What do we have? We have A. It's not an indication of the whole part of the ossicles. However, it is the first bone, second bone, and third bone. So remember, the first bone is the hammer. How do we remember this? It is the hairs. Hammer, anvil, stirrup. So the first one is the hammer. Right. And then B, semicircular canals. Right. And then C, the cochlea. D, the eustachian tube. E, tympanic membrane. Right, and then F, auditory canal. Right, now when we get to the questions, you already have the parts. You already know what the parts do, right? You know that the hammer has to vibrate as the tympanic membrane vibrates, right? Causes the anvil, the stirrup, to vibrate, causes vibration of the oval window into the inner ear, right? Semicircular canals, you know that those are involved in your balance, right? And your hearing. And then the cochlea, you know that when the fluid in the inner ear moves, it means that the cochlea will be stimulated. Not the cochlea, but the receptors in the cochlea, the organ of Corti, will be stimulated. Eustachian tube, you know what it does as well. The tympanic membrane vibrating auditory canal, you know that it also transmits impulses, the sound waves from the outer ear to the tympanic membrane. So here we have questions whereby it says give the letter and name of the part of the ear. Now, when you are asked to give the letter and the name, be sure to give the letter and the name. You can get the letter wrong and get the name right. You can get the letter right and get the name wrong. These marks are marked separately. So make sure that even if you're not sure about the other one, give the one a try. If you're not sure about the letter, write the name. If you're not sure about the name, write the letter. Again, you can write both. You have nothing to lose. Just make sure that you do not leave any blanks, especially on questions like this, where you just have to give a letter. You can look at any of these letters and say, okay, but I think the answer is C. Say C. Even if you're not sure whether C is the cochlea or the organ of Corti, say C. And if it's the organ of Corti and you said cochlea, then you would get a mark for that C. So A, it says allows air to move between the middle ear and the throat. That would be part D, which is the eustachian tube. Right, there it is. 1 mark for D, 1 mark for eustachian tube. Right. So D, eustachian tube. It contains receptors for hearing. Receptors for hearing is the organ of Corti. So that would be the cochlea. Cochlea is part C. Right. Remember, cochlea part C, eustachian tube part D. 2 marks. Is a membrane separating the outer ear from the middle ear. Now, you might not be sure of this, but if you are looking at this, you can see that this is the outer ear, and then we have the middle ear over here, and we have the inner ear. So separating the middle ear from the outer ear would be this part over here, which is part E. You're not sure if it's the tympanic membrane. You can simply say part E and get a mark for that. So it is part E, which is the tympanic membrane. 2 marks. And then is the first of the 3 ossicles. The 3 ossicles, hammer, infant, and stirrup. That would be the hammer. So your answer here would be hammer. And then the letter would be letter A. Conduct sound waves from the pinna to the structure labeled E. From the pinna to the part labeled E. So that would be this part here, F, which is the auditory canal. So you would have your marks for all of that. The cochlea, C, tympanic membrane or eardrum, E, A, the malleus or the hammer, F, the auditory canal or the ear canal. You can use whichever of these things that you prefer. Right. And then we have 2.4.2. 2.4.2 says, in a soccer match, the goalkeeper dived to save a goal from being scored by the opposing team. Describe the role of the semicircular canals in maintaining balance as the goalkeeper dived. Right. So here we are looking at what has happened. So here there was a change in position of the head, right? Because the goalkeeper dived. Right. Remember, when we look at balance, we look at the position of the head, or we look at change in direction and speed. Right. Now you are going to talk about how the receptors in the semicircular canals are stimulated by these changes, and how the impulses will be transmitted to the cerebellum. In this case, it's the cerebellum, not the cerebrum, right? And how the cerebellum then sends impulses back to your body in order for balance to be maintained. Right, let's look at that. So the cristae are the receptors that are in the semicircular canals, are stimulated by change in speed and direction. So as the goalkeeper dived as well, the speed also changes, the direction also changes as the head moves when the goalkeeper dives. Okay, so here we have your homework. The diagram represents the human male reproductive system. So you're going to look at the human male reproductive system again. Just give the labels from the seminal vesicle to the urethra. Give the labels again. Remember, there is really no use for you to label these different parts if you don't know the functions. So know the functions of each of these labels, right? Then you will be able to answer any question on the system. So if you know that, for example, part D transports sperm cells, then you would know what would happen if part D is cut, for example, or if part H is cut, for example. Now, if you do not know, and the question says, if this part is cut, then the male will produce semen without sperm cells, it means that you will not know that it is part D, if it's part D, or it's part H, if it's part H, because you only know the labels, but you do not know the functions. Right. Now, for example, part G, you should be able to understand why is part G structured the way it is structured. Why is part E placed where it is placed? What does it do? Then from there, you'll be able to answer any of these questions. Now, 1.4 here, we have questions whereby you just have to give the letter, not even the name, but just the letter. So if it says temporarily stores sperm, now you know all these labels, but you don't know their functions. It means that you will not be able to say, okay, part C is the one that temporarily stores sperm, if that's the case. So know the labels and know the functions of the different parts. So you have 6 marks, whereby you just give the letters. In the next exam, it might say give the letter and the name. Right. So you are going to go through that, and then the answers will be discussed in the next lesson. Now let's just have a quick summary of what we learned today. Know and understand the functions of the parts of the eye. Know and understand the functions of the parts of the eye. Don't just know the parts of the eye. Know them and understand their functions. Meaning that if we are looking at the lens, don't just be able to point at the lens and not know what the lens does. Know that the lens can change its shape for accommodation, right? Know that the suspensory ligament can become taut or loosened in order to have accommodation as well. Know that the ciliary muscle does this, the circular muscle does this. Don't just be able to label it. Know the function. Now, if your lens is not able to change its convexity, it means that your eye will not be able to accommodate the different distances, meaning that this individual will either be short-sighted or far-sighted. Right. Now, you don't know what the lens does. You know that this is the lens, but you don't know what it does. It means that you will not be able to answer questions when you are asked, what is the consequence if the eye is unable to change its convexity? Or what is the consequence if the suspensory ligament is damaged? We know that the suspensory ligament is the one that is going to control the convexity of the lens. Now, you don't know the function of the suspensory ligament. It means you do not know the consequence of the suspensory ligament not functioning. Therefore, you will not be able to give an answer based on that. So know the functions and understand them. That way you'll be able to understand how or what roles we play in processes such as pupillary mechanism or pupillary reflex, as well as accommodation. So this makes describing the processes better, right? And then next, all your drawings, is it a table, is it a graph, is it a drawing, or anything that says draw, it must have a caption, right? So you must be able to give that caption. Also, when you tabulate differences, if the question says tabulate, give it in the form of a table. Do not have a paragraph or bullet points that are giving the differences. Use it in the form of a table, because if you do not give it in the form of a table, automatically you lose that 1 mark for a table. So your graphs must have a heading, your drawings must have a heading. For example, we had the reflex arc. By simply saying reflex arc, that is a mark. So make sure you score those marks. Know the structure and functions of the different parts of the human ear, just like the eye. Know what the eustachian tube does. Know what the cornea does, the semicircular canal does, right? Once you know that, it means that you will be able to describe the whole function, the whole process of hearing. You'll be able to understand what would happen if the organ of Corti is damaged, right? What would happen if the tympanic membrane stops vibrating? What would happen if the ossicles are fused? What will the ossicles do? Now, if they are fused, what will happen? If the optic nerve Now, if we're looking at the eye, if the optic nerve is not functioning, what will happen? The auditory nerve, if it's not functioning, what would happen? Where are the impulses taken from when they move from the ear? If it's hearing, to the cerebrum. If it's balance, to the cerebellum. Understand all of these things, then you'll be able to answer questions much easier. So you can start step by step. Start by understanding the small things. Start by being able to label the structure. Now that you know how to label it, what does each part do? Now that you know that, take that knowledge and piece it together to understand the processes, to understand how the ear as a whole functions. If you know how the ear as a whole functions, it means that you can be able to understand what will happen if certain parts are damaged. How can hearing be lost? How can hearing be impaired? How can you then be able to use things such as the cochlear implants? Which part works as the microphone? Which part works as the transmitter, and so on, because you understand the different parts, right? Okay, I know how to differentiate the three neurons: the sensory neuron, the motor neuron, and the interneuron. Because if you were now drawing the reflex arc and you don't know the difference between these three, or which direction each neuron transmits impulses in, then you will not be able to draw that. You will not be able to give the direction, and therefore you would lose marks, because you might end up placing your motor neuron in the dorsal root or your sensory neuron in the ventral root, which then leads to you losing marks. So understand the little things, build up from that understanding, and then you will see that all of these things will be very simple, right? Now use your knowledge of the neurons to draw your reflex arc, then that makes it very simple, and you would be able to score as many marks as possible. Right, so this was our Gauteng paper 1 question 2 September 2021 paper, right? So that is the 50 marks that we were looking at. In the next lesson, we will be looking at question 3, which is also a 50 mark part. Section A, 50 marks. Section B, 100 marks. Question 2, 50. Question 3, 50. So that is how you'll be able to score your 150 marks in the exam. Okay, we have come to the end of our lesson for today. Thank you for watching. I will see you next time. Goodbye.